We have about almost a century now of this conventional model that we've been pursuing. And it's been leading us, things look good, at least on the surface, but when we get underneath the surface and see what we're doing to our environment, what we're doing to our health, we need to probably look for a different model. With what we're doing now, we're losing soils at 10 times the rate of formation. The thickness of a piece of paper is about 1 ton of soil on an acre. And we're losing at 10 times the rate of new soil formation, and in India and China, 16 to 18 times. The soils that we have left, about 40% of what we started with actually, we're losing through desertification, through soil salinization, pollution through chemical application and runoff, and also through urbanization. The water that we have, and you all know about water better than anybody here, of the 60% of the world's aquifers are being depleted faster than they can be refilled. And I know you have issues in this valley on that. The energy we used to use in agriculture. In 1940, for every calorie of fossil fuel energy, we would get 2.3 calories of food energy. Now, we get 1/10 of a calorie of food energy for every calorie of fossil fuel energy that we use. That's a 23 times reduction in efficiency, in energy efficiency in these systems. The fertilizer that we've been applying, see, agricultural land has not increased since 1955 very much, but our application of phosphorus and nitrogen has been 200 to 300 times percent more. Of the nitrogen and phosphorus we apply, only 10 to 30% of that gets utilized by the crop. The other 90 to 70% of the nitrogen and phosphorus you apply goes into our environment, goes into our rivers, our lakes, streams, our atmosphere. For nitrogen, the issues that we see, upper respiratory diseases, asthma, cancer, birth defects, cardiovascular disease, and sudden infant death syndrome. We see in Lake Erie a couple of years ago, the algal blooms. Yellow Sea in China, algae so thick you could float on it. The Baltic Sea, one of the most polluted bodies of water on the planet, and Lake Okeechobee. And then when they release Lake Okeechobee into the Gulf, they've destroyed the tourist season for the last 2 years with the red tides. We have about 110 documented dead zones on this planet, and they're all related to the agricultural systems that we're using right now. The herbicide resistance, USA is #1. We have 156 species of herbicide-resistant plants. We see also in our health. Autism in 1975, the chances was about 1 in 5,000 you'd have a child with autism. It's 1 in 59 now in 2018. It's not a causation, but there's an interesting correlation between our application of glyphosate to the autism rates. We see also in our foods that we're starting to get mineral depletion. Since 1940, all these elements have decreased in the vegetables and fruits and meats and cheese and the dairy that we eat. We've seen sometimes up to 80 and 90% reduction in the nutrient values of the foods that we eat. On farms, farm cash income is the same as it was in 1960. Farm debt has increased 14 times. Farm leverage is the highest level it's been since 1980. And we see return on invested capital in farms, we see a 3 times decrease in your return on the money that you invest in your farming operation. It's not a viable model. Systems can be destabilized with the least little thing. This is what we need to avoid in our ecosystem. You think this guy picked his check up at the end of the day? So we have challenges ahead. We need to produce higher quality and more nutrient-dense food on declining land area, using less water, fewer energy and natural resources, and do all this under difficult economic circumstances, with soils and ecosystems that are continually being degraded and in a system that is getting more unstable. Not a problem. We can fix this. But we're not going to fix it by keeping doing what we're doing. I think Einstein said that was the insanity, was keeping doing the same thing and expecting a different result. But Mr. Froehlich said it best: to change something, you need to build a new model that makes that model obsolete. So first of all, this is a living planet. Everything, the formation of this planet was all to do with living organisms. And they accomplished this by doing two things. There was a continual increase in system biodiversity as this planet evolved. And there's also an improving the efficiency and the capture of storage and utilization of energy we got from the sun. Looking over the last 4.5 billion years ago, water hit this planet about 4.2 billion years ago. Bacteria, about 4 billion years ago, very short period of time. Photosynthesis, anoxygenic photosynthesis, about 3.8 billion years ago. That started this whole planet on to what we see today. Photosynthesis was able to capture that energy, store that energy, and utilize it at a later time. We had virus precursors come along after that. They played in heavily in the changes in all these organisms. Nitrogen fixation, about 3.2 billion years ago. Oxygenic photosynthesis, here about 2.4 billion years ago, starting to lead to an oxygen-rich atmosphere. Viruses came into play to get into your prokaryotes, or eukaryotes, excuse me. And fungi came on the scene about 2.2 billion years ago. Ozone layer built up, mitochondria evolved. This little adaptation allowed a 16 times increase in the utilization of energy by these organisms. First single-cell animals about 750 million years ago, land plants 700 million years ago, Glomeromicata, the fungi that associate with plant and with bacteria, and then us. We got here, we are enjoying everything that has come before us. Some of the changes we've seen over the last 600 million years ago, due to the influence of biology or life. Right now, we are here. We've seen atmospheric CO2 concentrations up to 7,000 parts per million. We're now here at about 400. We've seen temperatures on this planet up as high as 25 degrees C. We're at 14 degrees C. We've seen atmospheric oxygen up and down through this whole period, up to 35%. We're sitting at about 20% now. All the influence of life on this, the first land plants were 700 million years ago. The Cambrian explosion, we had corals and shelled animals evolve in the oceans, starting to draw down atmospheric CO2. From the shelled animal, the calcium carbonate in their shells will be incorporated into the shell of that animal. Glomerulaceae, again, we're about 500 million years ago. Another little drop. We saw lignin, vascular plants, all evolved right here during the Silurian. Another big drop. As I say, each step along this path is an increase in the efficiency of these organisms to capture that energy and utilize it. Coral reefs have come and gone in different periods, but they also led to another drop in atmospheric CO2. 8-meter tall fungi, progymnosperms, roots, leaves, and seeds. This allowed plants to colonize the whole planet, not just to be around water bodies. A significant drop as we moved into the Carboniferous period. Is that better? Okay. And one of the reasons that we had such a buildup of coal in this era was there was no organism that degraded lignin. So all this plant material was just built up and built up. This is what we're enjoying today, all the fossil energy, all the energy that we can employ for building our buildings and moving around. Nature fixed that. A lignin-degrading microbe evolved about 275 million years ago. You see an increase in atmospheric CO2 from the respiration of the carbon in that organic matter. Conifers, ginkgos, and evergreens in here. Angiosperms evolved right about here. You can see another significant drop. And then a cooling period, landmass positioning changes. But most intriguingly, mammals or the ungulates and grasslands evolved in this period. We did not have grasslands until 55 million years ago, or grazers. And it was the action of those grazers and grasslands that built up soil carbon that we have been mining here for the last 100 years. Realize that as a living organism, we use almost every element on the periodic table. They're critical to our life, to all the metabolic processes that go on. In the Earth's crust, this is what each of the elements looks like as far as the concentration or the abundance. Interestingly, life, you are carbon, hydrogen, oxygen, and nitrogen. That's about 96% of the mass of your body. And all of those are gases. So the elements that you use out of the Earth's crust are all micronutrients, all used in small amounts. One interesting one, selenium, it's a very small concentration in the Earth's crust. It's about 50 parts per billion. You need it in your body at 200 parts per billion. Without it, you have problems with your immune function, viral infections, reproduction, mood, thyroid function, cardiovascular disease, and cancer. They have seen, when they canvassed the sediment in the oceans, there are 3 extinctions that we have no clue why they happened. But if you look at the selenium concentration in the seawater at that time, as they looked at the Earth's crust, there's 3 periods that it dropped below 1 part per million in the ocean. They line up very good with those extinction periods. They're suggesting that maybe it was a collapse of that food chain in that ecosystem. Us, as I say, we use almost every element. Iron is one of them that we use in our heme molecule. There's about 10 trillion of those made every second in your body. There's about 280 million of those in every red blood cell, and you have about 20 to 30 trillion red blood cells. The microbes are equally important. This one little microbe, Pseudomonas syringae, has a protein in its cell wall structure. That protein allows a supernucleation site. When that microbe is floating around in the air, it can condense a crystal of water on its cell wall that normally pure water doesn't freeze till minus 35 degrees C. With this organism, it can freeze at a minus 2 degrees C. So this little organism is instrumental in the hydrological cycle of this planet. Without this organism, we wouldn't have the amount of rain that we have, because dust only does it at, has to be minus 17 for dust to crystallize a water molecule. We have a large number here. It's not our national debt. That's about here. It's not the number of another second since life has inhabited this planet, or the number of sand particles on all the beaches of the earth. It is the number of biochemical reactions that go on in your body every second. You are flying. You have no concept of what's going on in your body with this rapidity of chemical reactions. So microbes are the backbone of every organism and ecosystem on this planet. You can see in the soils, stomach, root nodules. This is what makes life happen. This is how this planet developed. You have about 10 trillion human cells. You have 90 trillion microbial cells. You have 1 quadrillion resident viruses. All of these organisms work together to protect you. You have about 20,000 human genes. You have 2 to 20 million microbial genes. You are a very small component human. What do these organisms do for you? They digest your food. They do nutrient generation, cell renewal. We used to think they made up 60% of our immune system. We're now seeing it's 80% or greater. We see they control our appetites and cravings, vitamin synthesis that we can't get in our foods, detoxify carcinogens, prevent allergies, skin diseases, asthma. They can even turn on and off genes in your body that regulate brain development, anxiety, depression, autism, arthritis, and emotional behavior. So what happens when these communities are disrupted, either through administration of antibiotics or diet-related issues? Anybody recognize anything there? Almost every autoimmune disease that we have is related to a dysbiosis of our microbiota. We see in multiple sclerosis since 1950, type 1 diabetes, asthma have all increased 300%. Crohn's disease, 400% increase. Now, people can suffer for decades with Crohn's disease. And people have died from it. They do resections of intestines to try to cure them. They do more antibiotic regimes. But what they found out is if they can give these people with Crohn's disease another microbial community in their stomach with a fecal matter transplant, basically just inoculating their system with microbes from a healthy individual, they can see cures within 24 hours. When you look at our gut microbiota, the diversity compared to people in Tanzania, Malawi, and Venezuela, the USA, we have very low diversity in our stomach compared to those communities. When you look at an experiment done with mice to see, okay, we're going to take fiber out of the diet of these mice. What happens to that microbial community? The diversity, generation after generation after generation, decreases. You think, okay, we'll just bring the fiber back into the diet. That's this generation after generation, where they brought the fiber back. It does not come back. You have extinction events that occur in your stomach. And yet, when you come in with that fecal matter transplant, it's back and operating immediately. So if we can do this for humans, how about in the soils of our agroecosystems? Because we know we've destroyed the biology there with all the management protocols we've had, from the plowing to the biocides. Can we do this just by bringing the biology back into the system? Hope to show you that we can. First of all, we need to understand how nature is set up. There's a ladder of succession from a bare soil parent material up to an old-growth forest. And in that ladder of succession, we see increase in productivity. There's about 90 grams of dry biomass per square meter produced in a desert, compared to an old-growth forest at about 2,200 grams of dry biomass per square meter per year. As you see these changes, you also notice that we go from bacterial dominant systems to fungal dominant systems. This is very critical that we start to bring this back in our agricultural system because, well, I'll show you first. This is a bacterial dominant soil in California. It's a rice field. Just focusing layer after layer after layer. Those are all bacteria floating around there. So it's very bacterial dominant. This is what a healthy agricultural soil looks like. These are all bacteria. These are pieces of organic matter with a mesh of fungi grows on that organic matter. This is the old-growth forest, the Muir redwoods in California. You see very few bacteria. Lots of organic matter with fungal hyphae on that. This is the fungi. This is the highway. There are both logistics and communication. These are bacteria. This is real time. Bacteria moving along that thin film of water. These are your soils when they're healthy, having this system working. And fungi are the first thing to go on our farming methodologies. That's the first thing we destroy. So if you were going to start a war on an enemy, what's the first two things you want to destroy? Logistics and communication. So that's what we're doing to our soils, is losing these guys first. Where we are right now in agriculture, we're in a very bacterial dominant system. This is where we need to be, up here at about 1-to-1, fungal to bacterial ratio or higher. But we also need to understand to stay there on the ladder, agriculture is controlled disturbance. We need to disturb these systems. This was nature's way. The bison in the Great Plains here, they would be kept in tight herds by the predators. They would be allowed to graze about 40% of what was there. They would deposit their dung, their urine, their saliva, and they would be moved on by the predators. Now that dung and urine and saliva was actually with some fertilization to that, but there's also is a microbial inoculation, because that dung in a healthy system would be rolled into balls by dung beetles and placed in the soil and basically composting in place. So it's a great inoculation event as these organisms walked across the prairies. So can we mimic this in agricultural lands? This started out as a project with USDA. They said, do something with dairy manure. Do something good with dairy manure. And I started out with windrow composting. My wife got tired of me coming in with dirty clothes, and she helped develop this. She's the co-inventor. It looks simple, but you'll see it has powerful influence with very little application. We saw one of the biggest problems was the salinity on the dairy manure, and we moved the salinity down from 44 millisiemens down to less than 2 to 3. And it's also a vermicomposting process. The worms are part of this. But most importantly, it's a fungal dominant product that you end up with, and very biologically diverse. This is what it looks like when you're done. It's like a clay. You can squeeze it between your fingers. When we did analysis on it, this is just looking at the bacterial community, the change over the composting process from 160 degrees down to 60 through that time period, you can see there's a change. On our firmicutes, they start strong, but end up to be a small component. Likewise on others, like the proteobacteria, moderately strong, weaken off, and then come back strong through this process. Yes, sir?
Can you talk just, or maybe you're going to get into it, the temperature through the life of the compost, what it's likely to experience?
Sure. As we go through the composting process, that 160 degrees only lasts about 3 to 4 days in this bioreactor. Then you're done with the thermophilic phase. It will start to go down from there, down to around 60 degrees. Of course, here it's 32, right? And the worms are still pretty happy, it looked like, what we saw out there yesterday. What we noticed, these are two composts made a decade apart. Same process, different materials. This was made with dairy manure, 1/3, yard waste 1/3, and wood chips 1/3. This was all leaves. But you see a striking similarity in the structure, even though two different materials and a decade apart. So what we're saying is this composting process is the thing that's making this happen, because you're starting with two different carbon to nitrogen ratios, completely different materials, and yet you do end up with similar product. This is at 4 weeks what the bacterial species looks like with 80%. This is at 22 weeks. This is when most people will say, your compost is ready to use. If you allow it to go the year, this is what it looks like. You get a 4 times increase in the diversity of this compost. And that's, remember, one of the keys to survival on this planet is diversity. We had to demonstrate to USDA what the benefits of this compost were. And when I started out, I was an NPK junkie. I'm about 15 years reformed now. Because I thought it's all about the nitrogen, phosphorus, and potassium. But when we looked at the plant growth of our 2 composts compared to 8 others in our area, we saw an over doubling of plant growth compared. The nitrogen was practically nil. Phosphorus, the same way, a doubling of plant growth. The phosphorus didn't correlate to plant growth. Potassium also didn't correlate to plant growth. See, we're some of the lowest, and yet we had the best growth. We're starting to get around to, say, organic matter. That's got to be the key to this. It wasn't. What we did find out that did correlate was fungal to bacterial ratio. It was all about the biology. These plants were, they solely depend on the biology for their growth. So that prompted another experiment where we looked and see, okay, where is, of all the carbon that plant captures, where does it go in these systems? When we looked in a system that was bacterial dominant and low carbon, we were tracking the carbon that went into the root, the shoot, the fruit, the new soil carbon, respiration carbon, and also carbon that went into nitrogen fixation. And as we followed through, going from a bacterial dominant low carbon system to a fungal dominant high carbon system, we saw only 3% of the energy that plant captured go into the plant in these stressed soils, low carbon and bacterial dominant. Moving all the way up to a fungal dominant high carbon system, 56% of that energy went into that plant. Now, this is where we are in agriculture right now. We're about 11% efficient in this system. Theoretically, we should be able to go from 11% to 55%, a 5 times increase in the productivity on these lands. Does this work in the real world? Because that's all in a greenhouse. What we're promoting is what we're calling a biologically enhanced agricultural management, or BEAM. It's where we're coming in and inoculating. Yes, sir.
The last slide has been the focus of most of my interest.
Okay.
So, to me, my farm, I view it more as a, this is not a good way to make money, let me put that out there, but what I'm doing is not growing sweet corn or potatoes or wheat. What I see I'm doing is capturing solar energy, and I'm in the energy market. That's really, I'm competing in the energy market, not in beans or whatever.
Mm-hmm.
And so this is, where I see, we want to maximize that photosynthetic capture of carbon,
That's
etc.
Yeah.
But here we start to see it's more complicated. What do I want to do with the carbon that I might capture? Maybe one year I need to make money, so I need to concentrate on growing crop. Maybe another year I want to concentrate on growing soil. How easy will it be someday to manipulate soils to say, well, this is what I think I need to be concentrating on. I should shoot for this fungal to bacterial ratio because I know that's where I get maximum new soil. Or maybe that should be a cover crop, low expense, but I'm not trying to generate income from the cash crop. I'm trying to generate income in that I can build soil, and then the next year maybe, because it's a sweet corn crop, move back to this, that, and maneuver. And does that change throughout the season? Meaning in an annual crop cycle, how easy is it to maneuver between these pieces, and then do they stick?
Okay, well, realizing first that you are in an energy game is the first step forward. It's all about energy. And the second thing you need to realize, every chance you get to capture that energy, whether it's a cover crop or a commodity crop, you need to have something on that soil. That's what feeds the microbes. And if you're not feeding the microbes, you'll see as I go through this starts to build and stack on each other. You start to increase photosynthetic capacity of that soil. We're operating here, and we're not going to solve any problems here. At this rate of return, at this photosynthetic capture rate, we're not going to make any progress. We have to move up into this realm. It's like a bank account. You have to be making a certain amount of money and putting a certain amount of money in the bank to start building up your equity. And if you can't do that, you can't do it here. You have to have this improvement for this system to work for you, to be able to start putting money into the bank and energy into your system.
If I may jump in, yes.
Please do.
If you're looking at this part right here, this is where you're capturing the photosynthetic capacity above ground, right here, 56%. So if you improve your soil to that point, underground right here, that regardless if it's cover crop or cash crop, you should be able to maximize on your photosynthetic capacity, as long as you don't go back to high tillage and a chemical regime and all those detrimental practices that to destroy the microbial community. Does that make sense?
Yeah, yes. So maybe the data point I'm missing is kind of overall carbon capture. In my mind, it's not a sum zero game. So in my mind, I don't think I always want to be at 3.68. Maybe there's years or places where I want to maneuver within the economic reality between these four different goals. And
Well,
what I heard you say is no, just go to 368, and that's your best place to be.
no, this is just the range that we picked in the greenhouse. And the term manipulate, this is going to be a more observation part on you. You're not, the manipulation is what we've tried for over 100 years. And we need to start, we've spent 100 years trying to generate plants that can grow in poor soils, instead of focusing on the soil and seeing what the plant can do. And that's the place we need to go. Manipulating is what we've done for too long. And to see this system, this plant, why would it put 97% of its energy into the soil if there wasn't a reason? We need to follow nature on this system and not try to outguess her. Our key here is observation. That's going to be your job in the future, is to observe and see what you've done, what changes you make in your soil, and how it affects your crop.
Become adaptive.
Yeah.
David, I have a question with
Sure.
this slide too.
Sure.
So I work on rangelands, and I'm interested in using this to rehabilitate weedier areas to get it to transition to a more successional grassland. So when I was, reading some of Elaine Ingham's work about how weedier sites tend to have more nitrate versus ammonium, can you explain kind of chemically what's going on with the nitrogen fixation between the two different environments? And that conversion in the nitrogen
This
cycle?
I
I know it's probably $1 million question.
Yeah, and the perspective should be biology.
Yeah.
What is the biology doing? This nitrates, I'll show you at the end at Howard Buffett's farm, how we replaced over 170 pounds of nitrogen with 2 pounds of biology. So it's all about the biology. We need to stop concentrating on nitrate and nitrogen. Having the right biology there, this nitrogen can be fixed by these organisms. We need to move away, I think, from that NPK perspective, and even your micronutrients as well. These organisms make those micronutrients available, as I'll show you here, up to 1,000% increase in 20 months. So we'll get through this a little further, and you can ask that question again if it doesn't get answered in what you see in the data. Is that all right? Yeah. Okay.
Can I ask a question about the bank analogy?
Sure.
It sounds almost like building equity is putting energy into the soil,
Yes.
but what you get in return from the cash crop is almost like interest.
Yes, it's compound interest.
Yeah, so you're putting the money, you're putting time and energy to build the soil, but then what you're gaining is the interest in your cash crop.
And not only that, but you start building investment opportunities. You have the energy available for, if you have the microbes there, they can be busy making nutrients available for the next crop. It's like venture capital, is how I would put it.
That's a cool analogy.
And I say this system just starts building on itself. You start increasing photosynthetic capacity, that increases the soil biology. The soil biology starts putting more carbon in the soil. Now this is structured carbon. It's not amorphous carbon. It's carbon that's functional. So that carbon, in the form of the microorganisms, is busy mining that soil for nutrients and also storing nutrients. And then that enables you to have even more photosynthetic capacity. On the tail end of this, we also see a decrease in respiration in a healthier soil system. I'll show you here in a minute. And that allows you to build more carbon up because you're using carbon more efficiently. And that again allows you to have more photosynthetic capacity. And you just start stacking. And anybody that's adopted this, if you see Gabe Brown on the rangeland management, you don't need what I'm doing if you have cows. They can do it for you.
Right, but it's an issue of logistics.
Yes, yes.
You know, you can't high intensity graze huge expanses of grasslands. It's not it's a logistical problem.
Well, there's some ranchers that,
Oh, and
To do
I understand
Yeah.
that, but
this is a
a lot of
kickstart.
times we don't have enough production
Yeah,
to really allow that kind of management to occur without really affecting your bottom line of
oh yeah.
affecting animal weight gain and nutrition. That's a logistical issue with
And I'm
with
all
being
about
able
productivity.
To treat a landscape
and profitability.
exclusively with cows.
Yeah.
I
I'm
mean,
all
I'd
about
love to do it, but
I'm all about productivity and profitability on this.
Yeah.
I was in the private sector for 30 years before I got into academia. And I know these guys have to make a bottom line.
yeah.
And this offers a kickstart.
Right,
the
and my
system.
approach is, you know, how can, like, this is another tool in the toolbox.
Exactly.
If you're a range management specialist, you've got to use all the tools. Grazing, absolutely, we use that as a tool, but this could be another great tool to kind of address situations where you may not have a herd of 1,000 cows, or say you have a 100,000 acre ranch. Are you going to be able to, you know, use animal impact on every part of that ranch?
Well,
That's
hopefully.
one of the logistical issues.
Yeah, it is. It's having sufficient cattle to do the job, because it does take that disturbance. If you don't disturb that soil enough, you won't have that growth. If you disturb it too much, you set it back. It's all, as I say, it's an all observation game. And it's key that you are observing and you're learning along this process. There is no easy button. There's no formula. It changes every year. Nature is dynamic. We are linear in our approaches to everything. And it's tough with a linear mind to figure out how a dynamic system works. But this is one step towards that. So, does it work in the real world? Looking at what cultivated land can do now, they estimate from 600 to 900 grams of dry biomass per square meter. Seeing the old growth forest at 2,200 grams. Now we water it, we fertilize it, we weed it, and yet we can only get, what, a little over a third of what nature can do. With beam, beam and the transitional, we're seeing up to 2,200 grams of dry biomass per square meter. In an advanced beam, we're seeing about 3,200 grams, half again more than the most productive ecosystem on the planet, with the right biology. Now, we started out at 400 pounds per acre, and I'll tell you right now that's way too heavy. You can get results with much less than that, but this is what we started with because I had X amount of compost to go on X amount of land, and it was 400 pounds per acre. That's what it looks like on a square foot. It's a dusting. It's not a nutrient amendment. It is a microbial inoculum. This is after 1 year of beam where we went in and put the 400 pounds per acre on this right-hand side. And then we grew cover crops and a summer cover crop. There was 3 crops on this. Came back, planted the control, and this at the same time with this winter cover crop. Now, we saw after 1 year, it went from 50 grams of dry biomass per square meter to 250. That was a 5 times increase. But after year 7, and again, one application only, and just continually having a crop growing on this soil. Now this one, we were looking at putting everything back in. We were looking to see, we saw a carbon market developing. We wanted to see, well, what can a farmer do if they actually just put everything into improving their soil and growing plants and getting the carbon in. At year 7, we're seeing 1,141 grams. That's another 5 times increase after the first year. This is that same field. The summer cover crop, it was a black oilseed sunflower. It was a few vetches and sesbania, a forage soybean. This was, excuse me, June 23rd. 29 days later, it's about 5 feet high. This is 17 days later after that, over 7 feet high. Now, if you've grown black oilseed sunflower, they usually get 4 to 5 feet tall. They have heads like this. This is what they do on soils that have been regenerated. You can see the heads are almost as big as mine, and my wife says that's big sometimes. This is a sesbania crop. The first year we grew it was 4 feet tall. The second season was 6 feet tall. The third season was 12 feet tall. As I say, we don't know what these plants can do. We've always had plants bred for growing in a poor soil, and yet what happens when we change the soil with the plants that we already have? This was another experiment, and this was totally different from the others. I wanted a soil that had never had agriculture. I wanted no legacy issues from what chemicals had been applied. You can see that's where we live. It's the desert. This is a very sandy soil. On this, we inoculated it, we grew a cover crop, cut it, hauled it off, and planted again. Total extraction for 3.5 years. Now the first season, this cover crop was only 800 grams. This is the second season, 1,608 grams. This is the third season, 2,190 grams. Remember, the most productive ecosystem on the planet is 2,200 grams. This crop is planted in November. It idles through the winter. It wakes up about mid-February. We harvest at the end of April, so it has 10 weeks of growth. And in 10 weeks, we grew as much biomass as the most productive ecosystem on the planet. And also, you see 644 kilograms of nitrogen. That's about 700 pounds of nitrogen per acre rate that we fixed from the atmosphere. This is what it looked like through each season. This is our untreated, this is our treated. You see, we had almost a doubling of production as you go through those first 4 seasons. A whole year's production after that first season on the treated was over 3,000 grams of dry biomass. We saw about 3,100, almost 3,200 grams by the third year on this as that soil improved. As I was saying, that's 777 pounds of nitrogen, about 14.37 tons per acre of forage, a 5 times increase from here to there. So we were able to duplicate what we saw in the greenhouse in the field. That's the starting soil, just sand, no A horizon. The control, just growing a cover crop, is critical too. You got about a 3-inch A horizon. With the compost treated, we saw 7 inches. That much change. I'm a molecular biologist. I like to see who's doing what. We looked at those 3 soils, our desert soil, our control, and our compost treated, looking to see what organisms pop to the top. There's about 193 species that either correlated with the increase in plant growth or their population increase was 10 to 100 times more than any other organism on there. We saw about 2,750 species in this. And here's one bacteria that produces cellulose underground. It's actually growing organic matter itself. There's another one here that does 15% of the world's biologically fixed nitrogen. That's a Frankia species. This one right here is a radiation-resistant microbe. It can be bombarded with gamma rays to the point that it breaks up its DNA. It can reassemble its DNA and keep on going. These organisms can do some fantastic things. What we looked at is the different functions from nitrogen fixation, both free-living and symbiotic. We had both of those organisms, or both those categories, nitrogen cycling and carbon cycling. We have to realize for these systems to function, they're like our monetary system. If there's not a velocity of carbon and nitrogen flow through this system, it will collapse. It has to have a certain velocity to be healthy. Metal oxidation. We saw organisms in there that can extract almost every element in the periodic table out of the soil parent material if it's there. Phosphorus solubilization. We have over 40 years of phosphorus in our soil that we've applied. It's inaccessible to the plant because of the physical chemistry, and yet there are microbes that free that up. Antibiotic and antimicrobial production, biofilm and quorum sensing, go into that a little later. Organisms that degrade pesticides and antibiotics, and phytohormone producing. Now, why would a bacteria produce a hormone to increase plant growth? Unless it was in its best interest. These are the free-living and symbiotic nitrogen-fixing microbes we saw in there. You see several species, about 21 species. Nitrogen cycling, N2O degradation, carbon cycling. And you'll see a lot of these. This is one of the ones, a bug in a jug that you can buy. In bug in a jug, you'll get 5 to 7 species. In this, you get 2,750 species, and you can make it at home. Metal oxidation. You can see iron, manganese, chromium, boron, gold. These bacteria harvest all the elements that plant needs. Phosphorus solubilization. We had 10 species there. And again, Pseudomonas are huge in that, and you see them in the bug in the jug. Pesticide degradation. About 34 species that do well degrading the pesticides that we put onto our soils. What do they degrade? Malathion, chlorpyrifos, polyaromatic hydrocarbons, toluene, PCBs, halogenated hydrocarbons, 2,4-D. It's a good thing we have these microbes. That's why we don't have the buildup in our soils. Phytohormone production. If you're worried about food safety in that desert soil, E. coli, that's your marker microbe, was very strong. And yet as you bring this competitive inhibition back, you bring in the whole community, you see a decrease in that particular microbe. Quorum sensing and biofilm formation, there was 5 species that we saw in this. And interesting, this is a metatranscriptome analysis to let you know what metabolic functions are going on in that soil. These down here are housekeeping genes. They're genes that are essential for life to go forward. They're always transcribed at a certain rate. And there was only 2 proteins that were transcribed that were higher than these rates. Both of them were involved with quorum sensing. 5 to 10 times higher rates than any other transcription. Yes, sir?
Can you describe what is this quorum
Oh.
sensing?
Quorum sensing, they've seen that when bacteria get together in a community, they can do things, they can express genes as a community that they can't as an individual. Just like you as an individual can only get so much accomplished by yourself. And yet if you're with a community or with an organization that has a focus, all the talents come together and you can do things that you can't as an individual. And this is what happens in the biological world. Is when you get sick, that's quorum sensing. You get a group of microbes, get together, they start expressing genes that make toxins to your body. Well, it works in a beneficial way in the soils as well. So what are the benefits of regenerative agriculture? We're seeing that we're able to increase soil carbon. Now, most scientists that have been looking at this is 67 long-term no-till studies. These are agro-ecosystems with mulching. This is arable cropping systems. They're only seeing the ability to capture from 0.2 to 0.7 tons carbon per hectare per year. What we saw in B was 10.7 tons of carbon per hectare per year. That's a 0.25% increase in soil carbon per year. We see the potential to do a little over 0.5%, a 0.51% carbon increase per year. Now, this is what we saw, that 3.5 years, pretty consistent increase, 0.25% increase, 10.7 metric tons or 37 metric tons of CO2. This is what Gabe Brown saw. His first 13 years, he did no-till, crop diversity, cover crops. He saw only 0.1% increase in his carbon per year. And yet when he reached 3% soil organic matter, stopped using nitrogen fertilizers, and go into multi-species cover crops and livestock integration, he was doing over 0.5% a year. This is what his soil looks like now. This is what he's done with cattle. So I hope this kind of shows you what the potential is as far as how much you can change soils with cattle. We talked about respiration earlier. Now, This is back to that greenhouse test, looking what the respiration was in each of, from a bacterial dominant system to a fungal dominant system. Now we saw an 18 times increase in beginning soil carbon mass over this range. We saw a 5 times increase in microbial mass. That means there's more organisms there to degrade the carbon and there's more carbon available. This should have been exponential. So we only saw a 4 times increase in carbon respiration mass. So you start to get more efficient in this system as you go. This is Will Harris's. Looking at the years that he's practiced this regenerative grazing, at year 0, his respiration, and at year 20, when he moves from 0.25% soil carbon to 7.9% carbon, you see he saw the same curve I saw on respiration. He saw an increase in carbon use efficiency in the systems. That's what I was talking about, that stacking. You being able to hold the carbon longer because you're using it more efficiently. He had a 10 times increase in getting soil carbon mass and only a 4 times increase in soil carbon respiration as well. Flipping this around and looking at what percent of the original carbon in that soil was respired in a bacterial dominant system with low carbon, 55% of that carbon was respired. And yet as you increase the fungal to bacterial ratio, you can see only 11% of that carbon was respired. So your system is getting more efficient as you bring back this biology. On 10 times increase in soil carbon mass, a 4 times decrease in relative soil carbon respiration. This is how the Great Plains built up soil carbon. Most soil scientists look at this as a linear function, where more microbes, more carbon, more respiration. In every modeling for climate, biology is a black box. It's a linear function. It's not linear. You can see the system gets more efficient the more carbon and the more bacteria and fungi that you have. Looking at what happens with water, the first 1% increase in soil carbon, 5 times increase in your water storage capacity. You cannot lose by starting to bring carbon back into your soils. But bringing the biology helps you even more. We talked about the mineral depletion in our foods. This was the first 20 months of applying beam on that plot at Landecker. 5 sampling periods to look what happens to the micronutrients. We saw a 1,000% increase in manganese and iron, 83 to 40% increase in magnesium, calcium, zinc, and copper. We even saw increases in nitrogen, phosphorus, and potassium. Remember, we only applied the compost once, and then the rest of the time we're just growing plants. These organisms can bring all this back, and they can do it in systems that are actually moving away from availability. This is where the pH of our soil is about 8.2. Remember, iron and manganese increased 1,000%, and soil physical chemistry shows that they should be decreasing in availability. And yet with the right biology, these systems can make them available to the plant. Now productivity-wise, we saw cotton increase about double over what the conventional is in our area. We saw chili production almost double using the biology in the system with no fertilizers. Organic, they take a hit on their productivity because they're practicing conventional agriculture with natural nutrient amendments. They're not focusing on the biology. They could be doing this if they focused on the biology. So organic can be there. This is our cotton after a year and a half of being compared to 180 pounds of nitrogen per acre. You can see there's a little difference.
How old was that field that you started with for this test?
This one, old.
Yeah.
At the time?
Characterization of, you mean how long it had been in bean?
How long had the field been in cotton previously before you applied bean?
This field has seen everything. It's a university field plot. They've applied everything to it, so there's all kinds of issues with university land. our soils. That's why I did that other experiment on a non-legacy soil.
David, it wasn't cotton after cotton after cotton after cotton.
No,
after cotton.
no. They've done everything on these soils. I think this was our second year that we had gone into these plots, and Joe Ellington gave me one just to see what this would do. And this is what we saw after a year and a half of being. This was 2017, our cotton in July and August. And that's August 29th. We've got 6-foot cotton. Doesn't mean you have cotton, though, unless you do. We did about 5 bales of cotton, about 3,300 pounds of seed. That's a $2,000 an acre crop for cotton. No fertilizers, no herbicides. This is our chili. Patrick got some of the chili this last. The farmhands were waiting for ours to be done because they knew it had never been sprayed and they knew it never been fertilized. So, this is Howard Buffett's circle. He was kind enough to let us try this out on a circle in Wilcox, Arizona. We looked at a conventional and a beam plus 15% of conventional. In conventional, we use 256 pounds of nitrogen per acre. The beam, we use 38 units with 2 pounds of compost made into an extract and injected into the soil when we planted the seed. And this was beam only, just 2 pounds of compost extract injected. Now we had a sub-treatment in here of a multi-species cover crop on this side and a single species triticale. And you can see the difference in every treatment. Even the treatment with 256 units of nitrogen, the multi-species cover crop is essential to the success of these systems. If you can move away from single species, you're going to be doing better. But every treatment, we saw the multi-species all across the board do better. With 2 pounds of compost compared to a conventional, we matched productivity. First year, these were very compacted soils, low nitrogen, 5 to 10%, or 5 to 10 parts per million nitrogen in the soils. With the beam only, we only had a 6.6% yield. Now the farmer or the manager, when he looked at the corn, he said, "Why am I putting all this nitrogen down?" And we said, "We don't know. You should try the biology." And now they are. They built 2 bioreactors at Howard's farm, and they have several going in Africa as well. $88 plus an acre and $60 plus an acre on that. He was not profitable conventionally. We also, on the mixed species cover crops, this was the average production per square meter of dry biomass compared to the triticale. We had more productivity, and when you looked at the cost of the seed compared to your productivity, it was cheaper to do the 7-way mixes than it was to do the triticale as far as your return on your plant growth return on invested money. The Haggertys in Western Australia, desert type region, they are dryland farmers. They inject at about the same rate, about 2 pounds per acre. That seed is barely germinated. It's invested that much energy into developing that rhizosphere. You can see also on that. Now this, they get about 3 tons of barley per hectare in a region conventionally gets 1.5 to 1.6, just with biology. They've completely gotten away from any fertilizers. They come in, they're coating their seeds, they're injecting at 2 pounds per acre every planting, and they also do a foliar about 4 weeks into the growth. Now this is after they've harvested the barley. And this is the most telling to me of any of the slides or any of the pictures in this presentation. After they harvested the barley, they got a late fall rain. And in nice, neat rows, a C4 grass that they hadn't seen in 4 decades in that region came up. To have that seed bank last that long and know that it was simply the introduction of a biological community that allowed that system to re-establish itself was just phenomenal to me, to see the biology that strong. The Rainey Ranch, central New Mexico here, or south of us here, she has done this for about a decade, just grazing management, the multi-paddock grazing management, adaptive, I might add, and adaptive is the key. You can't do the same thing every year. You have to observe what's going on, how much forage you have, when to move the cattle. She's gone from 3 to 4 species of grasses to 45 in that decade. She reduced her medicine bill by 90%, reduced supplemental feed bills by 66%. She originally built swales to hold water for her cattle. They no longer fill. The first couple of years they did, but she improved her soil so much, the infiltration rate, that the water would go in and her aquifer actually started rising. This is Alejandro Carrillo, south of us in the Chihuahuan Desert. He sees the same thing. They all say it's about the soil biology. They all notice this, even though they're farmers and ranchers. He saw a 3 times increase of stocking rate compared to his neighbors, improved diversity of birds, improved native animal populations. This is what he started with. Lovely, huh? A lot of pasture there. 250 acres to support 1 cow. Now he can do 20 acres to 1 cow. This is what he's done in a little over a decade down there. So you can bring these soils back. You can bring these grasslands back. All around us, it was all grasslands when the Spaniards came. It was grass belly high to a horse, as they reported. So we can restore these. And it's all about, he says it's about the biology too. We were doing some research in the southeast U.S., looking at comparing adaptive multi-paddock grazing management to conventional. What we see, as you increase fungal biomass, you see standing crop biomass increase in these systems. That's not a bad correlation, 0.98. Now, same on the other end though, the bacteria, if you're bacterial dominant, it's negative correlation. So the more bacteria you have, the lower the productivity in these systems. This was the relative respiration. When we talked about these systems getting more and more efficient in using carbon, the natural areas, the respiration, compared to a conventional approach, compared to AMP. These systems, as I say, build on each other. You have to have the increase in photosynthetic capacity. You have to have the increase in carbon use efficiency, and that allows you to build your carbon up. So we can meet the challenges. We can rebuild degraded soils and increase soil fertility. We can produce more nutrient-dense food and forage. We can use less water to grow these crops by increasing both plant water use efficiency and soil water storage capacity. You can reduce the energy used by allowing nature to extract or fix essential nutrients. You can reduce both chemical pollution and atmospheric CO2 at the same time. And most importantly, you can improve your productivity and profitability in these systems. I've never seen farmers and ranchers happier than the ones that have adopted something like this, moving this direction. They know that they're making more money with less inputs. So why should we pursue this? There's no planet B, and there's not one close enough for us to get to. I'd like to thank the Buffett Foundation, Thornburg Foundation, Globetrotter Foundation, and Laird Norton Family Foundation for helping to fund this. They've been instrumental in moving this forward. And these are some of the links, if you're curious about the presentation or how to make the bioreactor, or how to make, I have a whole suite of YouTube videos to how to make an extractor on this. Any questions?